Hi everyone, welcome to your online lecture for the course Pathophysiology 1. In today's lecture we're covering Altered Cellular and Tissue Biology 2. It's important, or at least I recommend, that you have already gone through the content from Altered Cellular and Tissue Biology 1, as it will be a nice transition into today's lecture. Now before we get into the details of this topic, I'd like to review with you a normal cell. Remember that cells are the basic building block of life, and we're going to be talking about human cells, of course, throughout this course. But what does the normal cell look like? Well, the normal cell is going to have a nucleus, and the nucleus is going to be where the DNA is housed, which is the blueprint for what makes you, every detail about you. DNA also contains the instructions on how to make proteins. Now, proteins aren't just what you think of as what you consume as a protein. When I talk about proteins, I'm talking about the proteins that our body needs to produce in order to function. So, for example, sure, energy is an example, but enzymes are what your cells are going to be able to produce. Hormones. Our cells are also going to produce structural proteins that give structure to muscle, bone, teeth, skin, or even just structure to the cells themselves. Antibodies, that's another example. Hemoglobin. These are all examples of proteins, and the instructions for creating these proteins are found within our DNA. And then where are these proteins produced? Well, you might recall at the level of the ribosomes. And ribosomes can be found along the endoplasmic reticulum, so making this the rough endoplasmic reticulum, but they can also be found free-floating within the cell. Other important components of the cell include the centrioles and centrosomes that are involved in cellular division, the mitochondria, oftentimes several mitochondria found within the cell, and these mitochondria are going to produce energy in the form of stored ATP. And this great amount of energy that can be produced within the mitochondria is going to be dependent on the presence of oxygen. So in other words, our body needs to have oxygen present. It also needs to have glucose present in order to, or at least glucose is a preferred fuel source, but glucose and oxygen within the mitochondria, we undergo processes, Krebs cycle or the citric acid cycle and the electron transport chain, in order to produce the most amount of energy possible. A couple other structures. We have cilia, and this will come up throughout the course, the importance of cilia on cells. This is going to be very important for the movement of substances generally away from the cell. Lysosomes, another important organelle that contains very powerful digestive enzymes. So for example, these lysosomes can engulf pathogens or foreign substances that do enter the cell in order to prevent it from becoming harmful. We have the Golgi apparatus. This is where those proteins that were produced at the level of the ribosome will be really just packaged up and almost decorated or prepared for their, in order to prepare them for where their final destination will be. Then the smooth endoplasmic reticulum that you can see here, this is where the production of lipids occurs versus fat, or rather protein at the level of the ribosomes. And then finally, we have that phospholipid bilayer that surrounds the cell that really acts as a gatekeeper, permitting only certain substances to be able to enter or leave the cell, so it's a very controlled environment. But at the end of the day, there needs to be a good balance between production of substances within the cell and use. We'll learn that when cells are deprived of the things that they need to survive, that it can end up affecting not just single cells, but enough cells to affect the entire tissue and possibly even the entire organ. In Altered Cellular and Tissue Biology 1, we spoke about how human cells adapt under certain circumstances. But what happens when the adapted cell is constantly sustaining the stimulus that caused it to adapt in the first place? Well, cellular injury. And as you'll learn soon, cellular injury, it can be reversible, but it also can be irreversible depending on the extent of injury and the length of time that the cell has been injured. And when you have an irreversibly injured cell, it will die. So I'll remind you here of this diagram that we went over in our first class and how we have a normal cell here that under certain circumstances may have to adapt to a situation. And in this case, it's undergone hypertrophy, so it's gotten a bit larger, probably to be able to accommodate greater workload. And that adapted cell, if that workload is taken off, can then revert back to a normal cell. But if the stimulus remains, it will eventually become an injured cell. Again, depending on the extent of injury, it could be reversible. So if that heavy workload is removed, then it can reverse itself back to an adapted state, or in an even more ideal situation, back to a normal cell. But if that stimulus is not removed, that cell that is injured will eventually be in a state where the injury is irreversible and that cell will die. And you can see from this image that a cell that has died cannot revert back to either an adapted or normal state. Now, when this injured cell or adapted cell returns to a normal state, is it 100% normal again, free of any evidence that there was a problem to begin with? Well, not entirely. Usually there is some evidence, but for the most part, that cell can return to its normal functional duties. Now let's look at some examples of cell injury, and we're going to be talking about this throughout the next handful of slides before we start talking about a cell that is undergoing the process of dying. Cellular injury and cell death often result from hypoxic injuries. Hypoxia means insufficient or lack of oxygen within cells. And you need to be aware that hypoxia is the single most common cause of cellular injury. But in addition to hypoxic injuries, we have damage from free radicals, which we will talk about a little bit later, exposure to toxic chemical agents. We've given you some examples here. I'll point out carbon monoxide, which has been known to cause people to die because of exposure. Ethanol or alcohol as well is a toxic chemical agent that can cause cellular injury. And exposure to these substances, and of course lead, exposure to these substances will lead to cellular injury, and if the toxic chemical agent is not removed in time, then those cells will end up starting their pathway to death. Traumatic injuries such as a blunt, sharp object, gunshot wounds, asphyxia, which is strangulation, can all cause cellular injury. Asphyxia, for example, will just deprive the body of oxygen because of a traumatic event. Infections, injuries that cause or create a lot of inflammation, genetic alterations, nutritional imbalances. Physical agents such as temperature extremes, so being exposed to extreme cold or extreme heat. Hypothermia can lead to cell injury. And changes in atmospheric pressure. So for example, you've probably heard of the bends or decompression sickness, where if you have a diver that is deep within the ocean and then they decide they want to come up, they can't come up to the surface too quickly, or there can be this sudden change in pressures that can end up causing death. And finally, ionizing radiation, which we will elaborate on a bit as well. So we will talk about hypoxic injury, free radical injury, we'll look at ethanol and ionizing radiation on upcoming slides. And this here is just another image to show you the events of a normal cell either becoming adapted, injured, or dying. So it's just a different way of describing this image. How does hypoxia injure a cell? Well, ultimately by causing what's referred to as oncosis, which is really cellular swelling. So we're going to talk about the process of oncosis on this slide. Let's start with the image on your left. When a cell does not get enough oxygen, or enough oxygen can't reach the cell, we call this hypoxia. If you think back to cellular respiration from Anatomy and Physiology 1, and I won't test you on this component of normal anatomy, but when a molecule of glucose is transported into the cell, the first cycle we have is glycolysis, which occurs in the cytoplasm of the cell, and we know that that's an anaerobic process, and it does not require oxygen. Glycolysis literally means glucose splitting. It will form, and I'm just sort of summarizing here, it'll form pyruvate, and that process produces then 2 ATP. Now, if oxygen is present, pyruvate will then enter the mitochondria, and then the cyclic acid cycle or the Krebs cycle will begin, and this will produce another 2 ATP. Finally, we have the electron transport chain, and that's still occurring within the mitochondria, so oxygen needs to be present, produces upwards of 32 to 34 ATP depending on what you read. Now, if oxygen was not present because of hypoxia, so the cell's not getting enough energy, then that cell, it can't go through its normal anaerobic metabolism, so it shifts from aerobic metabolism to anaerobic metabolism. AT production is overall then depleted. So we have here hypoxia. I've explained the processes that would lead to then a reduction in ATP production. Why? Remember, not enough oxygen present to produce the maximum amount of ATP. Remember, back again to normal anatomy, the importance of the sodium-potassium pumps that regulate so many functions within our body. Now, in order for the sodium-potassium pump to function, it requires energy in the form of ATP. And so if this pump cannot operate properly, then you have a change in overall functionality. It's going to permit sodium to move into the cell. It's going to permit potassium to move out of the cell. This is not what we want. So also remember osmosis. Water likes to follow sodium. And so what will happen if you're getting sodium moving into the cell? Well, more water is going to follow. Now, this water accumulation in the cell is going to cause the cisternae of the endoplasmic reticulum to descend. And they descend, so here it's showing you distended cisternae of the endoplasmic reticulum. Cisternae is a flattened membrane disc of the endoplasmic reticulum, also of the Golgi apparatus. So again, water accumulation causes this distension, and then it can lead to rupturing. And then all of these ruptured areas are going to unite with other ones and end up forming these very large vacuoles. So what ends up continuing to happen is progressive vacuole formation, or we'll call it progressive vacuolation. And this will result in overall cellular swelling. The cell gets larger, it's distending, and this is called oncosis. It used to be referred to as, this is sort of an older term, hydropic degeneration. Sometimes you'll read it as vacuolar degeneration as well, but oncosis is the term that I'm using. Now, if cellular swelling affects one cell, not a big deal, but if cellular swelling affects all of the cells or many of the cells of an organ, then that entire organ will increase in weight because of this oncosis, and it will become very distended and very pale. And I'm referring to the entire organ. Organ increases in weight, it's very distended, and it's very pale in color because of this overall process of cellular swelling that affects an entire organ. Recall that cells form a tissue, tissues come together and form an organ. Let's now look at it in a similar way and add a few other important pieces of information. Before I start this slide, continuation of the previous slide, a couple of notes I want to make are the term understanding the difference between ischemia, hypoxia, and anoxia, which commonly students, and I've heard many people use these words interchangeably, but they do have different meanings, and I'll define them for you now. Ischemia is insufficient blood flow. to provide adequate oxygenation. So it has to do then with blood flow. We know that blood contains oxygen, so ischemia is you're not getting enough blood flow to an organ to give it oxygenation. And this in turn, if that cell is not getting the appropriate amount of blood flow, then it's not getting the appropriate amount of oxygen, and then that will lead to hypoxia of, well, probably not just a few cells, but to the tissue in general. So hypoxia is, as mentioned before, insufficient or reduced oxygen to the cells, to the tissue. Anoxia is absence of oxygen. Just like the term anaerobic, that prefix an is referring to lack of. So no oxygen, absence of oxygen within a cell is referred to anoxia, and those cells will die quite quickly. So let's put this together a little bit. Again, we're continuing on from the previous slide. If we have some sort of obstruction or cessation of blood flow, we've already discussed that this is referred to as ischemia, okay? Insufficient blood flow to provide adequate oxygenation. Well, what happens then? Well, that cell and all of the other cells are going to be having not enough oxygen in order to supply the mitochondria with what it needs to produce ATP. So this will overall create a reduction in ATP, because what we're really only getting now is we're in this anaerobic state, so we're producing only 2 ATP per molecule of glucose via glycolysis within the cytoplasm. So that only those 2 ATP, well, we're going to be using those up very quickly. Remember that our body's going to be using up 2 much faster than it would, up into the 30s that would be provided if oxygen was present. So what effect does this have? Well, we've talked about this part already here, that there is a reduction in the sodium-potassium pump function because it doesn't have ATP to function as much as it would need to. And that's going to cause intracellular sodium accumulation, extracellular potassium accumulation. To add into this, and this will be more relevant on the next slide, is the involvement of calcium. And in situations like this, we're going to get an influx of calcium within the cell, and that can lead to damage quite quickly. Then we know we're going to get an increase in water, because water is going to, according to osmosis, follow sodium in particular, which leads to cellular swelling. But just an extension off that, remember we're getting dilation of the endoplasmic reticulum, or the cisternae of the endoplasmic reticulum, and they can dilate so much. Remember we talked about the vacuole formation, that those ribosomes that are part of the rough endoplasmic reticulum can start detaching and not be able to produce the protein, because they're the protein factory, like they would have otherwise. Cellular damage then can lead to the deposition of lipids or fats within the cell and within the tissue. And we'll talk about lipid deposition when we speak about alcohol and its effect on the liver. Couple of more points. Hopefully it's pretty obvious that when we're in a state of not having enough oxygen, this anaerobic glycolysis is going to pick up. So if we're only producing 2 ATP from glycolysis, we need more. So what the body does is it takes the stored glucose, which is in the form of glycogen. So glycogen is stored glucose that's found in organs, for example, such as the liver. It takes this stored glycogen and starts breaking it down so that we have more glucose available. But if you remember back to normal anatomy, anaerobic glycolysis, one of the side effects to it is that it produces lactate. Normally when oxygen is present, we know that pyruvate that is formed from glycolysis will enter into the mitochondria and enter the different cycles there. But when oxygen isn't present, the pyruvate does not enter the mitochondria, and it will be converted into lactate. It undergoes a process called fermentation. This increase in lactate, you felt the presence of lactic acid in your muscles. If you do a real quick sprint, your body's using anaerobic glycolysis, and so you feel that acidity, that burning within your muscles, and that's that reduction in pH. So what we really end up having is this state of lactic acidosis. and our body can't clear it out as fast when we're remaining in this anaerobic glycolic state for such a long time because of the reduced oxygen availability. And then finally, one of the end events could be nuclear chromatin clumping. Chromatin, you might recall, is a massive genetic material that's composed of DNA and protein that condenses to form chromosomes in cell division. And if we're damaging DNA, well, you can only imagine how damaged that cell would be. Now, if that damaged cell or cells then undergo mitosis or cellular division, then we're reproducing a very damaged cell. So we'll talk about shortly what has to happen when our body does have damaged cells in order to prevent the possibility of major problems within our body. So why does all this happen? Why this isn't a daily event in your own body? Why would all of this happen? Well, there are so many different reasons, and that's why we're starting with this topic at the beginning of pathophysiology, because as we learn all of these different conditions that the human body can undergo, these conditions can lead to ischemia, hypoxia, and anoxia. So this gives you a general idea of what's happening with these diseases at the cellular level. So in this slide, we're going to be talking about calcium and how it can cause damage to the cell. Calcium ions are critical mediators of cell injury. In other words, they really do contribute to cell injury. Calcium ions are usually maintained at low concentrations within a cell's cytosol. The cytosol, remember, is that aqueous component of the cytoplasm of the cell. So it's where various organelles and particles are suspended. So calcium is usually maintained at low concentrations in this area. But with cellular damage, calcium continues to be released uncontrollably from various organs. For example, the endoplasmic reticulum can be damaged and release calcium. The mitochondria can do the same. And this free calcium, there's so much of it, and it can't be buffered or pumped out of the cell properly. One, because there's so much of it, but two, because the cell functions, its normal mechanisms that keep it in homeostasis are damaged. Now, what does this free calcium do? Many things. We're going to briefly talk about 4 main events. But what do these all have in common? All that are written in within the purple box is that this free calcium leads to uncontrolled enzyme activation. So uncontrolled enzyme activation will take place because of the presence of this free calcium, causing further and further damage to the cell. Remember, enzymes are going to generally end with a SE. That's how you can usually tell that it is an enzyme. So the examples that we have on this slide include activation of protein kinases, activation of phospholipases, activation of proteases, and activation of endonucleases, all that have very, very damaging effects on the cell. You are aware that the mitochondria of the cell is the powerhouse of the cell where ATP is produced, but another function that it has is to help regulate calcium levels. And so if the mitochondria has been damaged through the processes just discussed, then calcium is not regulated the way that it should be. We get this further increase in calcium or free calcium present within the cytosol. I also wanted to mention to you that we spoke a couple slides ago about vacuolation of the endoplasmic reticulum, but vacuolation will likely also occur in other organelles, including the mitochondria. So both the ER and the mitochondria become damaged by this vacuolation and extensive distension, leading to this increase in free calcium. Before I talk about just very brief information regarding these enzymes, I want you to make note that the uncontrolled calcium that is entering or infiltrating the cytosol of the cell is a very important and very oftentimes a final common pathway in many causes of cell death. In other words, think of this calcium infiltration as really occurring not long before a cell might die. Okay, the first enzyme we'll talk about are protein kinases. A protein kinase is a kinase enzyme, and kinase enzymes modify other proteins by chemically adding phosphate groups to them. So you might recall this process being phosphorylation. And so protein modification then can be occurring at a rate that is uncontrolled and at a rate that is very dangerous in the body. Next up are phospholipases. A phospholipase is an enzyme that hydrolyzes phospholipids into fatty acids and other lipophilic substances. And if you remember, the cell membrane is made up of phospholipids. So if phospholipids are being broken down, that's going to be damaging the cell membrane, which will make it much more permeable to the entrance and the exiting of substances in an uncontrolled and very concerning way. Activation of proteases. Proteases are enzymes that just break down proteins and peptides. And this can lead to cytoskeletal disassembly. Now, remember the cytoskeleton, this enables cells to transport vesicles throughout, undergo changes in shape, allows it to migrate. It's this very strong and dynamic structure that's formed by actin, microfilaments, microtubules, intermediate filaments. I won't ask you details. I just want you to be aware of what the cytoskeleton is, but with the activation of these enzymes that break down proteins, we're breaking down structural components as well of the cell. Finally, the activation of endonuclease. This is an enzyme that cleaves a polynucleotide chain by separating nucleotides. So an enzyme that separates nucleotides in a way that's not normal. And this leads to damage of DNA. Now let's go back to the different methods of cellular injury. We just spoke a lot about hypoxia and its role in damaging the cell. But what about free radicals? So that's our next topic. And a very important mechanism of cell injury is injury that's induced by free radicals, especially by what are called reactive oxygen species, or ROS. And there are many types of radicals, but those of most concern in biological systems are derived from oxygen. And so they end up being collectively referred to as reactive oxygen species. So first of all, know that what we're really talking about here is oxidative stress. So damage actually caused by oxygen, which sounds a little bit odd considering we said that it was lack of oxygen that can cause damage into the cell. But give me a few minutes to explain. First of all, what exactly is a free radical? A free radical is an electrically uncharged atom or group of atoms that has an unpaired electron. And having one unpaired electron makes a molecule very unstable. So those that are strong in or have a lot of education in chemistry would probably even understand this more than me. But this unstable molecule, in order for it to be stabilized, you're either going to be donating or accepting an electron from another molecule. When this attacked molecule that we're talking about loses an electron and becomes unstable, it becomes what's called a free radical. And a free radical is very capable of what's referred to as injurious chemical bond formation, which I've written here. Chemical bond formation with what though? Well, structures or molecules such as carbohydrates, proteins, lipids. So once again, these free radicals that become free radicals because they're attacked, they lose an electron, then they end up forming a bond with either proteins or lipids or carbohydrates in our body and causing damage. Our body, though, has this built-in system called an antioxidant system because it is normal for oxidative stress or free radicals to be present within our body, but our body has this built-in system that can combat it. But if free radicals happen in excessive amounts and overwhelm our body, then they can be very difficult to control, and they can initiate a whole chain of reactions that lead to oxidative stress all throughout the body. Free radicals are generated within cells in several ways. Just from normal cellular respiration produces free radicals. And we have an antioxidant system in place to take care of that. Absorption of radiant energy, the activation of leukocytes or white blood cells during inflammatory response. We'll understand inflammation more when we get to that topic. The metabolism of both chemicals and drugs can lead to free radical generation, among many other ways. Anything that we are exposed to that's toxic, and the many different poisons that we're exposed to on a daily basis within our environment can overwhelm our body with these free radicals and overwhelm our inborn antioxidant system. So antioxidants then prevent oxidation, and it's oxidation that causes damage within our body. It's almost like rust that can occur within our body. And so many foods that we eat, good quality fruits and vegetables and nuts and seeds, contain nutrients that act as antioxidants within our body. So people that want to boost their body's natural antioxidant system, they can do that better with a healthy diet. Some important examples here of reactive oxygen species that can form include superoxide, hydrogen peroxide, and hydroxyl radical. Now, what effects can this happen? Remember, it's excess ROS, or reactive oxygen species, that is damaging to our body, not just its presence in itself, because we can combat that most of the time, but that excess ROS, what can happen? Well, a few different things. These free radicals can cause damage via lipid peroxidation. What happens with this is we end up with destruction of polyunsaturated lipids, leading to membrane damage and increased permeability. Because remember, those cells, that phospholipid bilayer, is made up of lipids, and if it's damaged, then a membrane becomes much more permeable. Protein alterations, free radicals can cause fragmentation of polypeptide chains. So instead of these chains being strong and linked together, they can become fragmented. And this can lead to loss of proteins. It can lead to protein misfolding. So if proteins don't remain in their proper folded state, or they're folded not correctly, then it's not going to be an effective protein. Other things that can be affected are pumps in our body that are made of proteins. We have, remember, transport proteins that exist and are embedded within our cell membrane that help transport substances in and out of the cell. Alteration, or I should say DNA damage, will lead to a reduction in proteins that are produced, and that goes back to what we spoke about at the beginning of the lecture, because we know that protein synthesis is vital for really most essential functions within our body. And finally, mitochondrial damage. The mitochondria can be damaged by reactive oxygen species, which can then lead to the excess amount of calcium in the cytosol, which we know in itself, that excess calcium in the cytosol can lead to further damage of the cell. Now on to another topic of cellular injury, and that's ethanol. How does ethanol injure the cell? Well, the very basic answer is that it causes fatty changes or fatty degeneration. And this can happen within different cells in the body, but we're going to be speaking specifically of the liver. So ethanol, also known as alcohol, it is the number one mood altering drug within the US. It is so commonly utilized, and it does damage the liver. It reduces the ability of the liver to metabolize fats. So fats end up building up in the liver because they can't be properly metabolized, leading to fatty liver disease. And fatty liver disease is called hepatic steatosis. This condition, though, this fatty liver, is reversible if you stop drinking. But if there's prolonged consumption of alcohol, it can lead to scarring. So here we have a fatty liver, can be reversed to a normal liver. But if a person continues to drink, scarring can occur. And as soon as scarring occurs, then we have a cirrhotic liver, cirrhosis of the liver. And in Patho 2, there's a whole lecture talking about cirrhosis of the liver. Now in this example here, looking at the top, we're looking at a normal cell. And then we're looking at one that has intracellular accumulation of lipids found in hepatocytes. Hepatocytes are liver cells. And recall why the lipids build up. It's because there's abnormal metabolism. And that abnormal metabolism is from the overconsumption of alcohol. And so if metabolism is affected and we can't sufficiently remove substances such as lipids, then they end up accumulating within the liver. That was quite a lot of information on cell injury. But in response to significant external stimuli, cell injury can become irreversible, as we've discussed, and then cells are forced to die. We know that once a cell's injury becomes irreversible, that the cells will die, as just mentioned, and cell death has historically been classified as either necrosis or apoptosis, but they are different mechanisms. We'll first talk about necrosis and then apoptosis. Cellular death, oftentimes caused by hypoxia, will eventually lead to necrosis. Necrosis is really the sum of cellular changes that occur after local death, and it's the process of cellular self-digestion. So that's summarized here for you if you missed what I said. Self-digestion is also referred to as autodigestion or autolysis. But the next important statement to make that you need to be aware of is that cells die long before necrotic changes are evident microscopically. So if you see necrosis occurring, the cells have already died. Necrosis is not the first sign of cellular death. We spoke already about how when a cell is injured irreversibly, we will also see that membrane integrity is lost. And when membrane integrity is lost, the necrosis that has started within the cell doesn't just remain there. It now can leak out of the cell, and it can leak out of the cell and cause inflammation within the area. It can even affect other cells that weren't also injured, so it can almost spread. So we consider it then multicellular because it can affect many cells. Not that long ago, it was actually thought that necrosis was not something that's pre-programmed in our body, not something that our cells are intending to do, but newer research is suggesting that there are cells in the body that exhibit the ability to undergo this process of necrosis in a very almost pre-programmed fashion. So for example, programmed cell necrosis, meaning it's programmed into the cell to undergo necrosis under certain circumstances, is thought to be seen as a part of our innate immune response during certain infections. So you can see how research is changing all of the time. Different types of necrosis tend to occur in different organs or different tissues. And the five major types of necrosis that we will be briefly talking about are coagulative, liquefactive, caseous, fatty, and gangrenous necrosis. And we'll look at some images on the next slide. But before then, just some very brief information that you do need to know. Coagulative necrosis occurs primarily in the heart, kidneys, and adrenal glands, as you can see. Commonly results by hypoxia, and usually that hypoxia is caused by severe ischemia or lack of blood flow to that organ. A second common cause of coagulative necrosis, aside from the hypoxia from ischemia, is chemical injury from ingesting certain chemicals. Now, coagulation is a result of protein denaturation, which ends up causing the protein albumin, remember albumin is found within our plasma, to change from this very gelatinous and transparent state to form a very opaque color, be in a very opaque state. And the area of coagulative necrosis in the heart, for example, is called an infarct or a myocardial infarction. So clinically, obviously a very important type. Liquefactive necrosis commonly results from an ischemic injury as well, but to different parts of the body, to neurons and glial cells of the brain. Remember, neurons are nerve cells. Glial cells are supporting cells of our nervous system. So ischemia then to neurons can lead to liquefactive necrosis. But another example would be a bacterial infection. So we're going to be learning all about different bacteria, but staph, strep, E. coli, these types of bacterial infections can also lead to liquefactive necrosis. So what actually happens? What's the pathophysiology of this liquefactive necrosis? Well, it involves autodigestion of the tissue, which is part of necrosis anyways. Autodigestion of the tissue occurs and it causes the tissue to almost liquefy, causes it to soften and liquefy. And this area that's damaged ends up really being segregated from healthy tissues, oftentimes forming cysts. Now, if the cause of liquefactive necrosis was ischemia and therefore hypoxia to neurons within the brain, then this is called a stroke. Okay, next up is caseous necrosis. This type of necrosis is a combination of coagulative and liquefactive, so the two that we spoke about above, and it usually results from tuberculosis bacteria. Tissues, and so this would occur in the lungs, tissues end up resembling, almost looking like clumped cheese. And then finally, one of the last events to occur with tuberculosis is that this granulomatous inflammatory wall surrounds the area of caseous necrosis. Fatty necrosis. Fatty necrosis involves very powerful enzymes that break down fat called lipases. This type of necrosis occurs in the breast, the pancreas, and some other abdominal structures as well. Let's say somebody has injury to the breast, or they have injury to the pancreas, so they suffer, let's say, acute pancreatitis or inflammation of the pancreas. This can cause lipases to break down triglycerides, causing the release of fatty acids. And what happens with this release of fatty acids is that they end up combining with calcium, magnesium, and sodium, creating almost a soap, and it's called saponification. And so the necrotic tissue with fatty necrosis appears opaque and almost a chalky white. So once again, most common in the breast and pancreas, and it's usually if there's been injury to someone's breast, or if it's the case of the pancreas, somebody might be suffering an attack of pancreatitis or acute pancreatitis. Lastly, we have gangrenous necrosis. This results commonly from arteriosclerosis or the blockage of major arteries, most commonly seen in the legs. So if somebody has arteriosclerosis, then they get an infection, gangrene can result, and that can lead to amputation. So it's a very important consideration with diabetics, as you'll learn at the very end of the term. So continuation from the previous slide, here we have at the top left a myocardial infarction, or a heart attack, which would be coagulative necrosis. On the right here, we have a cerebral infarct or a stroke. You might recall that this is a type of liquefactive necrosis. Down here, we can see TB of the lungs, and this is a type of caseous necrosis. And then this is showing you gangrene. So those toes would be amputated. One more slide on necrosis. If a cell is plagued with irreversible injury, we know that cell will die. But this slide talks about some of the nuclear changes that can be noted. In some cells, the nucleus can shrink and become very small. It becomes this small, very dense mass of genetic material. This is referred to as pycnosis. Now, the nucleus will begin to fragment into smaller particles of almost like a nuclear dust, and this process is called karyorrhexis. Now, in the later stages of necrosis, most organelles are very disrupted at this point, and karylysis occurs. And this process involves nuclear dissolution and lysis of chromatin. I'll remind you from one of our previous slides that I told you that necrosis is the sum of cellular changes, so all the cellular changes that occur after local cell death, and the process of cellular digestion, which we know is called autolysis and autodigestion. And lysis is, you'll hear that term more than once in pathophysiology. This is just the disintegration of a cell by rupturing of the cell wall or rupturing of the cell membrane. Now on to apoptosis. Apoptosis is an active process of cellular self-destruction. It is considered and has been known for a very long time to be a type of programmed cell death. And it happens in not just pathological tissue, but also normal tissue. This is a fact that I won't test you on. The average adult may create upwards of 10 billion new cells every day and destroy the same number. What is that destruction process called? Apoptosis, programmed cell death. Programmed cell death, or apoptosis, is a very tightly regulated cellular program, meaning that there are specific mechanisms involved in the initiation and execution of apoptosis. So you can sort of think of apoptosis as being this very organized and cleaner event of cell death, where necrosis, not very organized, doesn't really follow very specific pathways that you would that apoptosis does, and necrosis is a lot messier. So what are some specific examples of apoptosis, both normal and pathological? So some normal examples include cellular death that occurs just normally during embryonic development, where certain cells are killed off that are unnecessary, and the thymus as well, which we know is a lymphoid organ that starts to really die off naturally as we get older. And a pathological example would be cancer. So when there's any, or even errors in cell division, then our body wants to make sure it can get rid of these damaged cells before our body continues to replicate them. Now, unlike necrosis, usually we have individual cells dying rather than it spreading to other cells, causing multiple cells to die. So I'll reword that to say that with apoptosis, neighboring cells are usually unaffected, unlike necrosis. Cells need to die. If not, we end up with this endless proliferation of cells, and we can form gigantic bodies within our body. Even if these cells are normal, they can become problematic. People that suffer from a low rate of apoptosis, this can allow for cancerous cells to proliferate or abnormal cells to proliferate. In this image here, you can see that we have cellular fragmentation occurring with apoptosis, forming these apoptotic bodies. And then our body is so clean with this process and programmed accordingly that we have these phagocytes that come in and are so kind to clean up the mess. And I didn't explicitly state it, but this blank here was just the point that I made about those that have a low rate of apoptosis. It can allow cancerous cells or abnormal cells to proliferate. At the beginning beginning of the lecture, I mentioned that ionizing radiation can damage cells. Ionizing radiation can be emitted by X-rays, for example. It is technically classified as a carcinogen, which is something that is known to have the potential to cause cancer. It's also classified as a teratogen, which is some agent or factor that causes malformation of an embryo. So that's why women are not X-rayed during their pregnancy to look at progress of the baby. Unfortunately, we had to learn the hard way from that with science, but now we know. But how does ionizing radiation damage the cell? We're not going into much detail at all. Just be aware that direct damage can occur, but indirect damage can also occur from the radiation. So we have here with indirect damage, we have water. Water will be ionized, so ionizing radiation. Water will be ionized and it ends up producing free radicals. And we know that free radicals are very damaging, and these free radicals in particular will damage macromolecules within the cell. It could be multiple different types of macromolecules that we know exist in our body, but in particular, what is very susceptible is DNA, or the nucleic acid. The cells in our body that are very particularly susceptible to damage include cells of the gastrointestinal tract, fetal cells, bone marrow, and lymph nodes. This slide is, well, the images are very outdated, but it just gives you an idea. You don't need to know any of these numbers, but in the early 80s, 15% of ionizing radiation came from medical use. Now, well, not now, in 2006, 48%, and theoretically even higher now. So we are now creating a lot of our exposure to ionizing radiation, and in many cases, it's unnecessary, and that's why we have to be very careful in the medical field to not expose somebody to ionizing radiation unless it's clinically indicated. Background radiation, ionizing radiation, just in case you're wondering, but you don't need to know the details. This is something that we're exposed to all the time. It includes radioactive material in the body, radiation emitted from substances such as soil, cosmic rays, various other things. Okay, on our final slide, we're going to talk briefly about what's referred to as dystrophic calcification. Within our body, calcium salts can accumulate within certain areas. It can accumulate in both injured tissue, but also necrotic tissue. And the damage really occurs from these calcium salts because they interfere with how, when they harden and they clump, they interfere with the normal cellular structure and normal function, not only of the cells, but of the organ itself. Dystrophic calcification is a type of pathological calcification, and as already mentioned, it can occur in areas of necrosis. Some examples of where it can be seen include those suffering from chronic tuberculosis of the lungs. It can be found in lymph nodes with certain conditions. Those with advanced atherosclerosis, remember that's narrowing of the arteries, usually as a result of plaque buildup. And heart valve injury. And here's an example showing you the buildup of calcium salts at the level of the heart valves. And as you can probably imagine, remembering back to the normal function of these valves, these calcifications are going to make it very difficult for these valves to function properly. The pathophysiology of this is actually not very well understood. You can see here just a very basic algorithm showing you the dystrophic mechanism of calcification, but when there's necrotic tissue, enzymes can be released. The release of enzymes can alter normal metabolic pathways, normal conditions, which can lead to the increased deposition of calcium within that area. And then the damage that the calcium can occur at that point could, in many cases, be life-threatening. That's the end of today's lecture. Thank you for listening.